MIT biological engineers have demonstrated a way to easily retrieve data files stored as DNA. This could be a step toward using DNA archives to store enormous quantities of photos, images, and other digital content. On Earth right now, there are about 10 trillion gigabytes of digital data, and every day humans produce emails, photos, tweets, and other digital files that add up to another 2.5 million gigabytes of data. Much of this data is stored in enormous facilities known as exabyte data centers. An exabyte is 1 billion gigabytes, which can be the size of several football fields and cost around $1 billion to build and maintain. Many scientists believe that an alternative solution lies in the molecule that contains our genetic information, that is DNA, which evolved to store massive quantities of information at very high density. A coffee mug full of DNA could theoretically store all of the world's data, says Mark Bates, an MIT professor of biological engineering. We need new solutions for storing these massive amounts of data that the world is accumulating, especially the archival data, says Bates. DNA is a 1,000-fold denser than even flash memory. And another property that is interesting is that once you make the DNA polymer, it doesn't consume any energy. You can write the DNA and then store it forever. Scientists have already demonstrated that they can encode images and pages of text as DNA. However, an easy way to pick out the desired file from a mixture of many pieces of DNA will also be needed. Bates and his colleagues have now demonstrated one way to do that by encapsulating each data file into a 6 micrometer particle of silica, which is labeled with short DNA sequences that reveal the contents. Using this approach, the researchers demonstrated that they could accurately pull out individual images stored as DNA sequences from a set of 20 images. Given the number of possible labels that could be used, this approach could scale up to 10 to the power of 20 files. Digital storage systems encode text, photos, or any other kind of information as a series of zeros and ones. This same information can be encoded in DNA using the 4 nucleotides that make up the genetic code A, T, G, and C. For example, G and C could be used to represent 0 while A and T represent 1. DNA has several other features that make it desirable as a storage medium. It is extremely stable and it is fairly easy but expensive to synthesize and sequence. Also, because of its high density, each nucleotide equivalent to up to 2 bits is about 1 cubic nanometer. An exabyte of data stored as DNA could fit in the palm of your hand. One obstacle to this kind of data storage is the cost of synthesizing such large amounts of DNA. Currently, it would cost $1 trillion to write 1 petabyte of data, that is 1 million gigabytes. To become competitive with magnetic tape, which is often used to store archival data, Bates estimates that the cost of DNA synthesis would need to drop by about 6 orders of magnitude. Bates says he anticipates that will happen within a decade or two, similar to how the cost of storing information on flash drives has dropped dramatically over the past couple of decades. Aside from the cost, the other major bottleneck in using DNA to store data is the difficulty in picking out the file you want from all the others. Currently, DNA files are conveniently retrieved using PCR, that is polymerase chain reaction. Each DNA data file includes a sequence that binds to a particular PCR primer. To pull out a specific file, that primer is added to the sample to find and amplify the desired sequence. However, one drawback to this approach is that there can be crosstalk between the primer and off-target DNA sequences, leading unwanted files to be pulled out. Also, the PCR retrieval process requires enzymes and ends up consuming most of the DNA that was in the pool. As an alternative approach, the MIT team developed a new retrieval technique that involves encapsulating each DNA file into a small silica particle. Each capsule is labeled with single standard DNA barcodes that correspond to the contents of the file. To demonstrate this approach in a cost-effective manner, the researchers encoded 20 different images into pieces of DNA about 3000 nucleotides long, which is equivalent to about 100 bytes. They also showed that the capsules could fit DNA files up to a gigabyte in size. Each file was labeled with barcodes corresponding to labels such as cat or airplane. When the researchers want to pull out a specific image, they remove a sample of the DNA and add that correspond to the labels they are looking for. For example, cat, orange and wild for an image of a tiger, or cat, orange and domestic for a house cat. The primers are labeled with fluorescent or magnetic particles, making it easy to pull out and identify any matches from the sample. This allows the desired file to be removed while leaving the rest of the DNA intact to be put back into storage. The retrieval process allows Boolean logic statements such as President and 18th century to generate George Washington as a result, similar to what is retrieved with a Google's image search.